Good morning. This is a revision of parts 2 and 3 of the molecular basis of inheritance. In this PowerPoint presentation, there are some slides on part 1. I'll be skipping them because part 1 is the basis for parts 2 and 3. So if you understand parts 2 and 3, then you've definitely understood part 1. This video will include only questions and answers, and it is based on the fact that you're supposed to have read the slides and seen the diagrams that are on the slides. Some of the questions might seem simple, but they are most of the time in free response questions. They are the introductory questions. So if you have a long free response question that consists of A, B, C, so these simple questions are usually letter A of the longer question. So the slides are 100 slides. We'll not be covering all of them. I'll be doing my best to cover the most important ones. So it's important to know what translation is. It seems easy, but unfortunately, most of the time you give an incorrect definition of translation, transcription, and all these processes. So translation is the process where we use a specific sequence of codons on a particular messenger RNA molecule to determine a specific sequence of amino acids in the corresponding polypeptide chain. So what is important in this definition is to know that every codon is translated into an amino acid and that codons are present inside a messenger RNA molecule, whereas amino acids are present inside a polypeptide molecule. So it is important to write it in that order. You use a specific sequence of codons on a particular messenger RNA to make or synthesize a corresponding, I cannot say complementary, because a sequence of amino acids cannot be complementary to codons. And translation thus means that you are converting the information on a messenger RNA molecule, messenger RNA molecule, into information on a polypeptide chain. Does messenger RNA provide information? Yes. Why? Because it includes a sequence of codons. Does a polypeptide involve information or include information? Yes, because it contains amino acids. It gives us the information about the type of those amino acids, the number of amino acids in the polypeptide chain, as well as the arrangement of those amino acids inside the polypeptide chain. So translation is the final step in the flow of information from DNA to the cytosol. What was the first step? The first step was replication to produce several copies of, or to produce an additional copy of itself. But most importantly, replication is important to allow cell division to occur, to allow continuity of the DNA molecule. But in the flow of information, it's not replication that is the most important step. In the flow of replication, the first important process, in the flow of information, is transcription that occurs in the nucleus, and then translation that occurs in the cytosol. The most important part of the definition, or the core of the definition, is the first paragraph. Now, what are the steps of translation? Of course, we all know that translation consists of three parts, or three processes, three steps, three phases, just like transcription consists of three phases. So we have the first phase, initiation, and then elongation, and then, of course, the third phase will be termination. But most importantly is to know how to define these processes. So initiation is where we start. It is the process by which we start the process of translation. And in order to give the signal to start the process of translation, we have to recruit methionine. Why? Because the first codon on messenger RNA, if this is the molecule of messenger RNA, the first codon on messenger RNA is AUG. And since AUG is the specific codon for the transfer RNA that carries methionine amino acid, then we are calling for methionine amino acid. Elongation means that methionine is not sufficient to make a polypeptide, so we must add more and more amino acids, which is the process of elongation. You have to differentiate, of course, between messenger RNA and the polypeptide chain that will be corresponding to that messenger RNA. So in the process of translation, we have a template messenger RNA, and we have a corresponding polypeptide chain that is made on top of that messenger RNA. Elongation is that we decide that methionine is not enough to make a polypeptide chain, so we must add more and more of these amino acids. And in order to elongate the polypeptide chains and to recruit those amino acids, we have to get their transfer RNA specific. And here it is very important, every time you talk about a transfer RNA, you have to mention the word specific, because each transfer RNA has its own specific amino acid. So elongation is the process by which more and more amino acids are recruited to the ribosome by transfer RNA. Recruited randomly, added in whatever sequence? No, it is according to the codon sequence, because every codon codes for a specific amino acid. That's why it is important to say that we are recruiting the amino acids according to a specific codon sequence on messenger RNA, and that these amino acids will be attached via peptide bonds, and that the formation of these peptide bonds is a process of hydrolysis, and it is catalyzed by peptidyl transferase. And peptidyl transferase, as we know, is an integral part of the ribosome. So again, I'm just erasing the editing. Let's read it again. Elongation is the process by which, every time you have to start by is the process. Of course, I haven't written them to minimize the amount of writing, but it is the process by which, or it is the process in which more amino acids are recruited. So this is the first part, recruitment of amino acids. By which molecules? By transfer RNA. According to what? To the codon sequence. Which codon sequence? The codon sequence on messenger RNA. Is it enough? No. We have to say that they are attached via bonds called, of course, they are covalent bonds, peptide bonds, and that this is catalyzed by peptidyl transferase, which is ribosomal RNA, or the ribozyme, and which is, and this is very important, an integral part of the ribosome. Termination is the process by which we stop the process of translation. How does termination occurs? Termination occurs when we reach a stop codon. You know that the ribosomal parts, the large ribosomal part and the small ribosomal part, move onto messenger RNA. And when they reach a stop codon, like what? Like UGA, like UAG, like UAA, there will be no transfer RNA. This is very important. There is no transfer RNA that is complementary to the stop codon. So what are we going to do if there is no transfer RNA carrying a specific amino acid that corresponds to the stop codon. Instead of that imaginary transfer RNA that does not exist, we will have a releasing factor. The ribosome will recruit a protein, and the releasing factor is a protein. And that releasing factor will play the role of the transfer RNA, and it will bind. What's interesting is that this releasing factor has a structure that is similar, although it's not an RNA, although it's not a ribosomal RNA, yet it has a structure that is similar, a 3D structure. I don't mean the chemical composition, a 3D structure that looks like a transfer RNA, and therefore it can sit onto the large ribosomal part and attach to the stop codon. This is very interesting to know that the releasing factor is a 3D molecule that has almost the same 3D shape, not chemical structure, as the transfer RNA. It will bind to here it's written that it binds to the translation complex, but before saying that it binds to the translation complex, you have to say that it binds to the stop codon. And when it binds to the stop codon, here, at that moment, the translation complex is complete, because the translation complex will consist of the ribosome, the releasing factor that looks like the transfer RNA, and the stop codon, in addition to the messenger RNA, of course. What this is the translation complex. Of course, there are other proteins that help this process of termination other than the releasing factors, but they are outside the scope of your curriculum. What is the result of binding, of transfer, of releasing factors to the translational complex? First of all, there will be separation of the two ribosomal units. And when the two ribosomal units become far from each other, then the messenger RNA will be released. And as we said before, the 5' end is where the ribosome fits first place, but it is therefore the first part of the messenger RNA molecule that is released, because the ribosome moves like this, and the last part of the messenger RNA to be released is the 3' end. So first of all, this termination occurs by separation of the ribosomal subunits, release of the messenger RNA, taking into consideration that the first part to be released is 5' and the last part is 3', and finally the release of the polypeptide chain. The release of the polypeptide chain is our ultimate goal, because we want this polypeptide chain to be released into the cytosol, because this polypeptide chain will form a protein. Essentially, the process of translation occurs in the cytosol, but the polypeptide chain was imprisoned inside the translational complex. Now it is released into the cytosol. It will form a protein. That protein might be a functional protein like an enzyme or a hormone or an antibody, or it might be a structural protein like collagen, like elastin, or it might be a motor protein like myosin. You have a multiplicity of proteins that are important in the cell and that are being released through the process of termination. Now, the third question is, how does translation in the prokaryotes differ from translation in the eukaryotes? So here we have a very short comparison. Of course, prokaryotes are unicellular organisms that do not have a true nucleus, but eukaryotes are unicellular or multicellular organisms that possess a nucleus, and therefore they possess a nuclear membrane. And this is very important for the differentiation. Now, in prokaryotes, there is no nuclear membrane to separate between the nucleus and the cytosol. So all the ribosomes inside the prokaryotes are cytosolic, meaning that there are no, furthermore, there is no endoplasmic reticulum in prokaryotes. So I cannot say in prokaryotes that there are free cytosolic ribosomes and that there are attached ribosomes that are attached to the rough endoplasmic reticulum. Why can't I say this? Because essentially there is no endoplasmic reticulum. So all ribosomes are exclusively cytosolic and free inside the prokaryotes, whereas in eukaryotes, some ribosomes, of course, are free and cytosolic, whereas the others are attached to the ER, the endoplasmic reticulum. And when they are attached to the endoplasmic reticulum is described as being a rough endoplasmic reticulum. Now we all know, and we come again to the process of to main difference between eukaryotes and prokaryotes, that here in eukaryotes we have a nuclear membrane. We all know that the process of transcription of messenger RNA occurs in the nucleus, and that the process of translation of messenger RNA into a polypeptide occurs inside the ribosome in the cytosol. So in eukaryotes, it's essential to say here that the messenger RNA is first transcribed in the nucleus, and then after being transcribed in the nucleus, it is processed in the nucleus by removing the introns and then splicing the exons together, and by protecting the messenger RNA and helping it to attach to the ribosome using the 5' cap, and then by protecting it again by adding the polyadenine tail. And we might be seeing this later on over the slides. You know that the messenger RNA at the beginning of transcription in eukaryotes is long, and that it contains exons and non-coding introns, and then exons again. So the exons are coding, they code for amino acids. The introns are non-coding, they do not code. So this is immature messenger RNA, unprocessed messenger RNA, and it is essentially longer in eukaryotes than in prokaryotes. In eukaryotes, it contains introns, but in prokaryotes, no, because the genome of the prokaryotes is too short to carry introns. It cannot afford having non-coding areas. So what we do in eukaryotic genomes, that we first remove the introns that are non-coding, and then we splice or attach the exons together. In such a way, the messenger RNA becomes shorter, and this is what we call processed RNA, messenger RNA. Afterwards, we add a polyadenine tail at the 3' end to protect it, and a sequence here that is called the 5' cap to protect it and to help it attach to the ribosome. All this processing, in addition, of course, to what we've discussed before of alternative splicing of messenger RNA at the end of the first presentation, all this is protective to the messenger RNA and occurs in the nucleus, so that in eukaryotes, at the time when messenger RNA is released from the nucleus to the cytosol, it will become mature RNA. In prokaryotes, we don't have this long story. Why? Because in prokaryotes, messenger RNA can be translated while it is transcribed. Why? There is no separation. It's all cytosol. There is no separation. So this is the DNA. We transcribe it into messenger RNA, and the ribosome is just there in the cytosol waiting for the messenger RNA to be transcribed on the spot. We do not need processing because there are very, very few introns. So in prokaryotes, the process is much simpler. Messenger RNA can be translated while still being transcribed because there is no nuclear membrane and there is no messenger RNA processing. How does the alternative flow of information affect translation in retrovirus. Of course, at the first while, you can see that this question is a very difficult question. What is alternative flow of information? The normal flow of information is, in order to explain what is the alternative flow of information, we have to say what the original flow of information is. The original flow of information is from DNA. This is the classic flow from DNA to RNA. So the alternative flow of information would be the reverse. Instead of starting by a DNA template and ending by transcribing messenger RNA, in reverse flow of information, we will start by RNA and we will form a molecule of DNA. And in order to differentiate between this DNA that has been produced by the alternative flow and the DNA that was the template in the classic flow, we call this DNA complementary DNA. There are only a few organisms that can do this. There are only a few organisms that can do the alternative flow, and they are called the retroviruses. Why? Because essentially the retroviruses do not have a DNA genome. They have an RNA genome. And they want to convert that RNA genome into a DNA genome for one simple reason, just in case their host cell has DNA genome. So if they want to infect that host cell and they want to insert their viral genome into the host genome, it's more appropriate to have a DNA genome, because the DNA of the virus would be much easier to attach or anneal to the DNA of the host. So it's a necessity for retroviruses, this group, or this famous group of viruses, to convert their RNA genome into a DNA genome. So they need a very specific enzyme of transcription. It's not an ordinary messenger RNA transcriptase or an RNA polymerase, sorry. It is a reverse transcriptase enzyme. Why? In order to be able to use their RNA to transcribe complementary, again, the C is very important, from their own DNA. And then after having obtained complementary DNA, the issue is not yet resolved. Why? Because this complementary DNA that has been produced by the reverse transcriptase enzyme is single-stranded. And I remind you that the reverse transcriptase enzyme is one of the most important enzymes, and that this enzyme is frequently encountered on the tests. So afterward, the complementary DNA has to be replicated by a DNA polymerase. This would be therefore the template, and we produce the replicate or the complementary strand. And at that moment, it can be incorporated or intruded into the host genome. This viral DNA has been first transcribed, and then it has been sorry. This second part, this second paragraph, starting here at the red mark, is talking about the fate of that double-stranded. We have finally, the virus has finally succeeded in converting its RNA genome into a double-stranded DNA. And the purpose of converting it into a double-stranded DNA was clear from the beginning. The purpose is to intrude it into the host genome. So the host genome, say it is, let's say that it is a human genome. So in such a case, it would be a linear genome with linear chromosomes. So this viral DNA that I'll be drawing in blue will be incorporated into the host genome. So this would be the viral genome. Of course, it might be incorporated at the beginning, at the end, or even in the middle of the host genome and causing a mutation. Why? Because whether it is whether it is attached at the beginning, the viral genome, and this is the host genome, whether the viral genome is intruded at the beginning or at the end or in the middle of the host chromosome, it will cause a disruption of the host genome. And if it disrupts the host genome, then it will result in a mutation because you have changed the sequence of nitrogenous bases on DNA. And this means what? This mutation will be shown when this DNA that has been intruded, this viral DNA, is transcribed and then translated by the host cell, by the host metabolism, by the host machinery or biochemical pathways. But in such a case, the host cells will be producing viral proteins and not host proteins. And this is the reason why, for example, some people who get hepatitis C virus might end up by having cancer liver. This is because of this insertion. And by the way, whatever kind of inserted viral genes that stay dormant in the host genome, and by dormant, by the way, I don't mean that they are not transcribed and translated by the host machinery, but I mean that when I say a dormant viral gene, I mean that it is not shifting to the lytic cycle that ruptures the cell. Staying dormant means that it will not lyse the cell, but it is directing the host machinery to produce its own DNA, and this is what we call the prophage. So what is the prophage? The prophage is a dormant virus that is intruded into a host genome. Dormant does not mean that it is not harmful. Dormant does not mean that it did not cause mutation. Dormant does not mean that it will not be translated and transcribed into viral proteins inside the host DNA and cause mutation and cause cancer. Dormant means only one thing, that it will not be shifting to the lytic cycle and rupturing the host cell. The fifth question is, how does the process of translation show evidence of common ancestry? Of course, the process of translation is using a sequence of messenger RNA to form a corresponding sequence of polypeptide. Of course, the sequence on messenger RNA is a sequence of codons. So what do we need to do this? We need a codon, a specific codon on messenger RNA. We need a transfer RNA with a specific amino acid, and this transfer RNA should be having an anti-codon that is complementary to the codon. We need, of course, a ribosome. We need releasing factors. So if I tell you that the genetic code that is represented on messenger RNA is universal, meaning that all codons code for the same amino, or I mean that every codon codes for a specific amino acid, and this rule applies similarly in all living organisms. For example, if in all living organisms, the AUG codon will code for the amino acid methionine. So it's the same rule in all living organisms, whether eukaryotes or prokaryotes. This is what we explain as the genetic code is universal. Nearly all the organisms use the same genetic code, and the translation mechanisms are nearly the same in all living organisms. The nucleotides used and therefore the codes on DNA and the codons on messenger RNA are common. Not only this, but there is more advanced compatibility, which is that even the viral DNA molecules and the viral RNA molecules are compatible with the host genomes. What do we mean by this compatibility? We mean that although this is a virus, yet its DNA, if intruded inside a higher host genome, a eukaryotic genome, the eukaryotic genome will be able to transcribe and translate this foreign genome, which is the viral genome. Now, the proteins of replication, I'll skip that slide because it's part one, and the description of the function of each of I'll skip all the slides that are related to number one or part number one. Of course, you'll be using them at the end of the year for the final test, inshallah. Now, the translation of the viral genomes introduced into the bacteria or into eukaryotic genomes is possible. This question number 11 is related to what we have just said in the previous question, that the genetic code is universal. The question says what? The question says that if you introduce a viral genome into a bacteria or into a eukaryotic genome that is totally different from the viruses, yet it can be translated again and again because the genetic code is universal and the same code codes for the same amino acid in nearly all living organisms. Now, here there is a very important question that is talking about, or that is asking about, the functions of ribosomal RNA. We have talked before about ribosomal RNA in the classroom, and we have said that there is another name for ribosomal RNA, which is the ribozyme, and that ribosomal RNA studies have shown for the first time in the history of sciences that ribosomal RNAs are not only carrying codons like other types are not only carrying information like other RNA molecules, but they carry out a structural function and an enzymatic function. You might be wondering about what I've just said now, that the ribosomal RNA might have a coding function. Yes, but this is a more advanced level of information. It has only a few coding information to allow the ribosomal RNA to say or to know where to bind to the ribosome. But we will skip this part because it's outside the scope of your AP curriculum. But ribosomal RNA unexpectedly has structural functions and enzymatic functions. The structural function is that the ribosomal RNA, together with some polypeptides that are translated in the cytosol and that are called the ribosomal polypeptides, share in the formation of the material of the ribosome. They are the building molecules of the ribosomal large subunits and small subunits. And we have seen before during our study of the cell that those two ribosomal subunits are assembled together in the nucleolus of the cell. And this was a very important function of the nucleolus, meaning that the final assembly of the ribosome is inside the nucleus, but the ribosome has to leave the nucleus. Why? Because the ribosome works where? It works in the cytosol. So after having been assembled in the nucleus, it will leave the nucleus through the nuclear pores and reach the cytosol, where it will help in the process of translation by hosting the molecules of translation like messenger RNA and transfer RNA. But ribosomal RNA also has an enzymatic function, and it is sometimes called catalytic RNA or catalytic ribozyme. It catalyzes what? It catalyzes synthesis of polypeptides. So inside the large ribosomal subunit, this ribosomal RNA that entered in the structure of the ribosome is not only structural, it's also functional. It acts as an enzyme. And it's actually what we have been naming for years peptidyl transferase. For years and years, I've been teaching this and saying to my students that there is a peptidyl transferase enzyme that attaches the amino acids together by making peptide bonds between them. Actually, it is breaking down the bond between the transfer RNA in the P site of the ribosome, this elongated peptide chain, and it is breaking it here and transferring it to the next transfer RNA that is in the A site and that is also carrying a series of amino acids. So if you're not mastering that part, you can just say that it attaches the amino acids together by dehydration synthesis and forming a peptide bond between them. But if you're mastering it, you will say that it breaks down the because you know that the ribosome has three parts: an exit part, and then a peptidyl part, and then an amino acid or an amino acid transferase part. So here you have the transfer RNA that is carrying the growing polypeptide chain, and here you have the transfer RNA that is trying to, or that is bringing the newly coming amino acid. So actually we break down that bond and we translocate that peptide chain to be attached to the newly coming amino acid. Of course, you can review the diagram on your PowerPoint presentation to remember this. Now, what are the salient features of each of the steps of translations? What are the most prominent features of each of the steps of translation? In initiation, we have the transfer RNA carrying methionine amino acid that binds to the first initiation codon, which is AUG. In termination, which is the last step, this is a recap of what we have said. We reach a stop codon. No more amino acids are attached because there is no transfer RNA. Translation stops, polypeptide is released. And when we reach the stop codon, there is no further translation. In elongation, we are bringing more and more amino acids according to the sequence of codons on messenger RNA, and each newly arriving transfer RNA carrying a new amino acid will enter the A site on the ribosome, waiting for the polypeptide chain to be detached from the transfer RNA on the previous site, which is the P site, and they are added to the growing polypeptide chain using ribozyme or the peptidyl transferase. Of course, every transfer RNA should have an anti-codon that is complementary to the codon, and this is a recapitulation of the three steps of translation. We will skip the slides on replication. Now we will talk about the energy cost of translation. Every time you are adding a new amino acid, you are consuming a molecule of energy, which is guanosine triphosphate. So one guanosine triphosphate is hydrolyzed into, as usual, like in ATP, into guanosine diphosphate, inorganic phosphate, and energy is released. And this energy is enough to recruit the amino acid for translation. So energy is essentially used for recruitment of the amino acids, not for binding them. At this stage, we are talking only about the energy needed for the transfer RNA to recruit the amino acid from the cytosol. I would like to add something about the catalytic activity of ribosomal RNA. You might be wondering how ribosomal RNA can act as an enzyme. As I told you before about the transcription factor, that it might acquire a 3D shape that makes it similar to the usual transfer RNAs. Here we can say that the ribosomal RNA can also have a 3D shape. And in that 3D shape, so here this is a very important point to emphasize. It's not only a protein that can shape itself into a 3D functional protein with active sites and catalytic sites and active domains, but the RNA, especially the ribosomal RNA, can shape themselves into 3D shapes. You might be telling me, but the transfer RNA has a 3D shape. Yes, but it is a 3D shape that does not have a catalytic site. But in ribosomal RNA, the ribosomal RNA arranges itself in a 3D shape, although it's not a protein, that is similar to the active domain or the active site on an ordinary enzyme. Since we are talking about translation, then we are talking about the genetic code, because what is a genetic code? The genetic code is the sequence of triplet codes on DNA, or the sequence of triplet codons on its deputy, which is messenger RNA. So if you're talking about the chief genetic material, which is DNA, we say triplet codes. What is a triplet code? It is a sequence of three sequential nitrogenous bases on DNA. But since messenger RNA has codons that are complementary to the triplet codes, on DNA we say code, on messenger RNA we say codon, then the sequence of codons on messenger RNA is also a genetic code. So why is the genetic code redundant? Because we have only 20 natural amino acids, but we have 64 triplet codes. Since we have 64 triplet codes on DNA, then we have 64 possible codons on messenger RNA. So the number of codons is much, much greater than the number of amino acids, although we say that every amino acid, or every codon, sorry, we should start here, every codon codes for a particular amino acid. So we are expecting, since the number of amino acids is 20, that the number of codons is 20. The fact that the number of codons is 64 means one simple thing, that it's not only one codon that represents the amino acid, but I might have 2 or 3 codons that code for the same amino acids. And this is what we call redundancy of the genetic code. This means simply that the genetic code is larger, looser than, it's just like when you are wearing an oversized t-shirt, for example. So the genetic code is oversized just because many amino acids have more than one codon. So for every specific amino acid, there will be more than one codon in addition to the 3 stop codons. Why do many when I say many amino acids, I mean that this happens frequently. This happens for many amino acids, but actually every amino acid, or most of the amino acids, have more than one codon to represent them. I'll stop here, and then I'll be sending another video.